friends, welcome to my video. In this video, we are going to see
types of culture media.
At first, we are going to see
what is culture medium.
In microbiology,
Medium means
any substance used to provide nutrients for the growth and multiplication of microorganisms. Any population of microorganisms cultivated in a medium is known as culture. We learnt the meaning of the terms medium and culture. Now let's see what is culture media. Culture media means any substance that support the growth of microorganisms. It is also known as growth media. Culture media contains nutrients, growth promoting factors, energy sources, buffer salts, minerals, metals, and if it is a solid medium, then it contains solidifying agents. Next, we are going to see about types of culture media.
Based on the physical state of the media, culture media are classified into liquid media, semi-solid media, and solid media.
Based on the chemical composition of the media, culture media are classified as simple media, complex media, synthetic or defined media, semi-defined media, and special media. Special media are further divided into enriched media, enrichment media, selective media, indicator media, differential media, Sugarmedia, Transportmedia, Assaymedia, and Anaerobic Media.
We learned what is culture medium and what are all the types of culture media. Next, we are going to see some important details about each media. Let's start with
liquid medium. Liquid medium is a nutrient containing liquid culture medium used to cultivate or maintain the stocks of microorganisms. Liquid medium do not contain a solidifying agent, so this medium remains as liquid. Let's see some important advantages and disadvantages of liquid media. Liquid media are convenient for growing a large number of bacteria very quickly. In liquid media, bacteria are free to move about. Bacteria growing in liquid media may not exhibit specific characteristics for their identification. It is difficult to isolate different types of bacteria from mixed populations using liquid media. A good example of a liquid medium is the nutrient broth. Nutrient broth. Nutrient broth is a nutrient containing liquid medium used for growing bacteria. It is also known as broth medium. The basic ingredients of nutrient broth are peptone, meat extract, sodium chloride, and water. Next, we are going to see about solid media. Solid media is a solid culture media containing nutrients and a solidifying agent. Because of the presence of solidifying agent in solid media, the media remains solid in room temperature. Solid media contains ingredients which support growth of a wide range of microorganisms. In solid media, bacteria multiply at the site of inoculation and form clear visible colonies with distinct colony morphology and exhibit many other characteristic features such as pigmentation, hemolysis, etc. so that identification is made easy. Solid media is also used for the isolation of bacteria as pure culture. The most common solid media is nutrient agar. Nutrient agar. Nutrient agar made by adding 2% agar to nutrient broth. The main difference between nutrient agar and nutrient broth is that nutrient agar contains a solidifying agent and remains solid in room temperature. broth do not contains it and remains in liquid form. Next, we are going to see about semi-solid medium. If the concentration of agar is reduced to 0.5% or less, semi-solid or sloppy agar is obtained.
Semi-solid medium is useful for the cultivation of microaerophilic bacteria,
for the determination of bacterial motility, etc. Here, one should note one important point, that is, increasing the concentration of agar to 6% prevents spreading or swarming by organisms. Next, we are going to see some important points about agar. Agar is universally used for preparing solid media. Agar is extracted from the cell wall of some species of red algae. Agar contains polysaccharides, inorganic salts and small quantities of a protein-like substances. It has no nutritive value and also not affected by the growth of microbes. The unique property of agar is the large difference between the gel forming temperature and melting temperature. That is, agar melts at 85 degrees Celsius and solidifies at 32 to 40 degrees Celsius. Agar gels are reversible and translucent. These properties makes agar an ideal solidifying agent for microbiological culture media. Next, we are going to see about classification of culture media based on chemical composition. At first, we are going to see about simple media. Simple media is a culture media containing nutrients which supports the growth of most non-fastidious bacteria. Example, nutrient agar, nutrient broth, etc. Next, we are going to see about complex media. Complex media are a type of media composed of rich or complex nutrients, but the exact composition is unknown and hence the media is called complex media, which means complex media have some complex ingredients such as extracts and digests of yeast, meat, plant, etc. The exact chemical composition is unknown. So, we are calling it as a complex media.
Complex media is also called as undefined media. Let's see why we are calling complex media as undefined media.
The presence of extracts from animals or other microbes makes a media undefined, as the entire chemical composition of extracts are not completely known. So, we are calling complex media as undefined media. Example, brain art infusion agar,
Tryptic soy broth,
etc. Next, we are going to see about synthetic or defined or chemically defined medium. Synthetic media are prepared from pure chemical substances and the exact composition of the medium is known. These are used for various special studies such as metabolic requirements. Synthetic medium is also known as defined medium because we know the exact composition of the medium. A chemically defined medium is entirely free of animal, plant or microbial derived components. Defined media can also define as media composed of pure ingredients in carefully measured concentration dissolved in double distilled water. Next one is semi-synthetic media. When the composition of the medium is approximately known, are termed as semi-synthetic medium. Example, simple peptone water medium. Next, we are going to see about special media. Special media are used in the identification of bacteria and are supplemented with dyes, pH indicators, antibiotics, some essential nutrients, growth factors, etc. Special media are further divided into enriched media, enrichment media, selective media, indicator media, differential media, sugar media, transport media, Assaymedia, and anaerobic media.
Before saying about the special media, we are going to see what is fastidious organism. Fastidious organism is any organism that has complex or specific nutritional requirements. These organisms will grow only if specific nutrients are added in its medium. Example, Neisseria gonorrhoeae, which requires blood or hemoglobin and several amino acids and vitamins to grow. Next, we are going to see about enriched media.
The term enriched means to make rich or richer. Some organisms are more exciting in their nutritional needs. To support the growth of these organisms, substances such as blood, serum, egg are added to a basal medium. This medium is known as enriched medium. Enriched media contains growth factors, vitamins and other essential nutrients to promote the growth of fastidious organisms. Example, blood agar, chocolate agar, egg media, etc.
For your easy understanding, we can also define enriched media as follows. In order to support the growth of fastidious organisms, we are making a medium more richer and we are calling that medium as a enriched medium.
Next, we are going to see about enrichment media. Let's see the meaning of the term enrichment. Enrichment means the act or process of improving the quality or power of something by adding something else. Just to try to understand the meaning of the term enrichment. While I am explaining what is enrichment media, you can follow it easily. Okay, now let's see what is enrichment media. Enrichment media. An enrichment medium contains some component that permits the growth of specific types or species of bacteria because these specific organisms alone can utilize the component from that medium. In mixed cultures, the bacteria to be isolated is often overgrown by the unwanted bacteria. Unwanted means non-pathogenic or commensal bacteria. For example, if we want to isolate Salmonella typhi from feces, Salmonella typhi being overgrown by Escherichia coli in cultures from feces. In such situation, substances which support the growth of bacteria to be isolated or substances with an inhibitory effect on those unwanted bacteria are incorporated in the medium. The important point to be noted here is that
by using this medium,
we are actually improving the power of bacteria by adding some substances which support the growth of bacteria to be isolated or substances with an inhibitory effect on those unwanted bacteria. So, we are using the term enrichment media. Example, tetrathionate broth, where the tetrathionate inhibits coliforms while allowing typhoid and paratyphoid bacilli to grow. Another example is selenite F broth. Enrichment media is a liquid media. We are using the enrichment media before culturing in order to increase the number of the organisms to be isolated and eliminate the unwanted organisms.
Next, we are going to see about selective medium. If you are able to understand what is enrichment medium, then it is easy for you to understand what is selective medium. Let's see what is selective medium. If the inhibiting substance or enhancing substance is added to a solid medium, then it is known as selective medium.
While explaining what is enrichment medium, I told you enrichment medium is a liquid medium. If we add solidifying agent to that liquid medium, then we will be calling it as a selective medium.
So, selective medium is a agar based medium which supports the growth of specific or selected organisms by restricting the growth of other unwanted organisms. The selectivity of the medium depends on the addition of antibiotic agent, chemicals, indicator dyes and media with altered pH. Example, deoxycholate citrate medium for dysentery bacilli.
Next we are going to see about indicator medium. Indicator medium contain an indicator which changes color when a bacterium grows in them. For example, incorporation of sulfite in Wilson and Blair medium. Salmonella typhi reduces sulfite to sulfide in the presence of glucose, and the colonies of Salmonella typhi have a black metallic sheen. Next, we are going to see about differential medium, a medium which has substances incorporated in it and used to visually differentiate different types of microorganisms based on their different colony color or a change in the color of the medium. Example, McConkey's medium, which consists of peptone, lactose, agar, neutral red and taurocolate. Let's see about McConkey agar. McConkey agar is a selective and differentiating agar that only grows gram-negative bacterial species. It can differentiate the gram-negative organisms based on their lactose metabolism. The fermentation of lactose produces lactic acid. Acid decreases the pH of the agar. McConkey agar contains pH indicator that turns pink under acidic conditions. Therefore, lactose fermenting gram-negative bacteria will form pink colonies and non-lactose fermenters will form off-white opaque colonies. This may also be termed indicator medium. Sugarmedia. In microbiology, any fermentable substance is denoted as sugar. Example, monosaccharides, polysaccharides, alcohols, etc. The usual sugar media consists of 1% of the sugar in peptone water along with an appropriate indicator. A small tube called Durham's tube is kept inverted in the sugar tube to detect gas production. Sugar media are used to determine whether or not a bacterium can utilize a sugar. Next, we are going to see about transport media. Transport medium is a non-nutritive, balanced and buffered medium designed to preserve the viability of bacteria during transport and also to minimize bacterial overgrowth between the time of collection and sample processing. Example, Stewart's medium. Next, we are going to see about Assaymedia. Media are used for the assay of vitamins, amino acids and antibiotics. Example, antibiotic Assaymedia. Finally, we are going to see about anaerobic media. Strictly anaerobic microorganisms require the absence of oxygen and a low redox potential to initiate growth. Anaerobic media are used to grow anaerobic organisms. Example, Robertson's cooked meat medium. Friends, I am concluding this video here and see you soon in my next video. Thank you friends.